Good afternoon and welcome back to this next session. In this session, we are going to be focusing on diversity in clinical trials, and we have 5 presentations. We actually have 7 speakers because the last presentation will be a triumvirate, and we very much look forward to the speakers. I'm going to introduce these speakers to you in aggregate right now, and then allow them to succeed one after the other in their presentations. So the first speaker will be Manish Satarangani, and Manish will be addressing us on the topic of pediatric clinical trials. He is very well qualified to do so as the director of the Vaccine Evaluation Center at the BC Children's Hospital Research Institute, and he's an associate professor in the Division of Infectious Diseases in the UBC Department of Pediatrics. His training was at Oxford, Cambridge, London, and he's worked with the Oxford Vaccine Group, and we very much look forward to his presentation. The next speaker will be Professor Gina Ogilvy, who will be addressing the topic of underrepresented and marginalized minorities in clinical trials. I have the privilege of working regularly with Professor Ogilvy. She's a Tier 1 Canada Research Chair in Global Control of HPV-related Diseases and Prevention, and a professor in the School of Population and Public Health, and has done many large trials on vaccination and screening in British Columbia and low and middle income countries globally. The third talk will be presented by Mr. Jim Pankovich. He will be addressing ethnic factors in multi-regional clinical trials. Mr. Pankovich has more than 25 years of experience in the biopharmaceutical industry with extensive strategy development, clinical study management, and operational experience, particularly as relates to the development of novel compounds and taking these novel compounds from clinical development right through to NDA filing. And we very much look forward to benefiting from his experience as well as his knowledge in this regard. The next speaker will be Dean Emeritus, John Cairns. John is speaking to the topic of older adults in clinical trials. He is a cardiologist, and his research is focused on antithrombotic therapies for acute ischemic syndromes and atrial fibrillation. He has led many large national and international clinical trials and has published over 240 papers as a consequence of this. He's the former Dean of Medicine at the University of British Columbia, Faculty of Medicine, and he's also past president of the Canadian Academy of Health Sciences, and we very much look forward to hearing from him. The final presentation will be by 3 speakers, led by Dr. Julia Bickford, who's the Regional Director of Research for Evaluation, Research and Analytics at the Northern Health Authority, and is also the co-lead of the BC Support Unit at Northern Centre. She's had an opportunity to work with a variety of stakeholders to develop capacity and infrastructure in the North, supporting a high-quality, evidence-based healthcare system, and we very much look forward to her presentation that she is actually sharing with Dr. Nadine Caron and Victor Dinenko. Many of you will know Dr. Nadine Caron. She's a member of the Sagamok Anishinaabe First Nation. She's a practicing surgical oncologist in northern British Columbia. For many years, she had been the sole Indigenous physician within BC Cancer, and she is an Indigenous scholar within UBC's Faculty of Medicine. Most recently, she was named as the inaugural First Nations Health Authority Chair in Cancer and Wellness, and she currently leads the development of the Northern Biobank Initiative, which is a partnership with the First Nations Health Authority, and we very much look forward to benefiting from her experience and knowledge in this regard. And finally, Victor Dinenko is the Research Project Manager for the University of Northern British Columbia and works with Dr. Caron on the Northern Biobank Initiative as the Program Manager for Infrastructure and Implementation. His PhD is in molecular biology, but he has been extremely effective and instrumental in developing infrastructure and capacity in Northern Health Sciences research, and is a key member of the Northern Biobank Initiative team. So that is our layout for this afternoon, and I very much look forward to hearing from all of our speakers, and I thank you for being part of the audience.
Afternoon. My name is Dr. Manish Sadarangani. I'm a pediatric infectious disease physician at BC Children's Hospital and director of the Vaccine Evaluation Centre, where I lead a number of pediatric and adult vaccine trials. I'd like to thank the organisers for asking me to speak to you this afternoon on the subject of pediatric clinical trials. And I would like to pose the question of whether or not this is an expensive luxury or a fundamental necessity. These are my disclosures. So I want to start off just by taking a high-level overview of what is translational research. And around 10 years ago, this is a paper that was published defining various types of research. So basic research as being performed without thought of practical ends, clinical research as being conducted with human subjects or tissues, including epidemiologic and behavioral studies, and outcomes and health services research, and translational research of applying discoveries in the laboratory to development of clinical trials in humans and enhancing adoption of best practices in the community. And just picking up on this final point about translational research, I think if we're planning on applying our discoveries to children, then we need to be doing our research including children. So do we need pediatric clinical trials? So these are some reviewer comments that I've had from grant applications in the last few years. So are they just an expensive luxury? Some negative comments, so designing, implementing and funding multi-center clinical trials is very difficult in the current Canadian context was one reviewer's thought on it. How easy is it to convince the ethics committee to obtain blood samples from otherwise healthy infants? Not at all from my experience. And I hope that most of you are aware that we do trials in healthy infants all the time, and the ethics committee, as long as it's done appropriately, is completely open to that. And finally, whether or not it's a fundamental necessity. I'll come back to this a bit later, but I want to highlight this comment that understanding is hampered by lack of suitable animal models capable of providing insight. And if we want to produce drugs and vaccines for humans, then we need to have human studies. So let's start off by thinking, who is young? So this is from the Merriam-Webster dictionary, and as well as the usual definitions you might expect, intriguingly also includes a definition of being a desirable age for use as food or drink, or maybe desirable for use in clinical trials. Why is this population important? So this just is from the Global Burden of Disease study. They have a number of publications in the Lancet over the last few years, and really highlights the death rate here, shown on the y-axis, across the different ages, and the death rate in children, particularly in young children, is much, much higher than most of the rest of the population, with the exception of the other end of the population as the elderly. And a lot of the trials that we do are in this middle portion here of healthy adults or even unhealthy, but in adults and young adults, but you cannot apply that to children. They're a very different population. My research is on infectious diseases and vaccines, and so a lot of that is really tied in very closely with the immune system. And this graphically nicely shows the dynamic changes happening in the immune system over the age spectrum. And what you can see is that most of these changes happen in the pediatric age group. And if you take a preterm baby, that's different to a term baby, different to a 1-year-old, it's different to a 2-year-old. You have a period of relative a static phase, if you like, and where there's not a huge amount of changes in the immune system over late childhood and adult life, although there are still changes happening. And then the next big changes occur in older adults, and as people get older over the age of 60 onwards, sometimes known as inflammaging. And so this, not only is it important if you want to design interventions for children to look at children, but it's important to get the right age group as well. So going back to the comment about animal models, the old adage is that animal models lie. I just have one example of that to show you, and this is an experimental HIV vaccine that was developed several years ago now. There's various different formulations that were tried in the non-human primate models. This is as close as you can get to a human model. This one in the bottom here showed that with an adenovirus 5 viral vectored platform, there was complete suppression of HIV infection with this experimental vaccine. And interestingly, this platform is being tried right now for COVID vaccines. When this vaccine formulation was then trialed in humans, there was a clear difference between the vaccine group and the placebo group. And if you look in some of the subgroups, particularly those who already had high levels of antibodies against the adenovirus 5 vector, or interestingly in uncircumcised men, again, hugely significant differences between the vaccine group and the placebo group. But if you look closer, you'll see that the vaccine group here in the dashed line and the placebo group here is in the solid line. And on the y-axis is the HIV incidence. So what we're seeing here is, yes, there's a huge difference, but actually those who received the experimental HIV vaccine were more likely to get HIV infection than those who received the placebo. And on the basis of this being a harmful intervention, this trial was stopped early. So completely contrary to what was found in the animal models. As well as animal models lying, I think when you think about kids, so do adults. So in the early 2000s, there was a meningococcal disease epidemic in New Zealand. So it's a devastating disease, 10% plus fatality rates in children and adults, very high rates of long-term neurological disability. And so a vaccine was fairly rapidly, maybe not quite as rapidly as COVID vaccines, but anyway, nevertheless, a meningococcal vaccine was very rapidly developed and trialed across different age spectrums, because again, this was an epidemic going across the population. And what was found that in adults, 96% of people responded to this vaccine, whereas in children, it was around 75%. If you looked at the antibody levels, again, adults had good antibody response with a antibody concentration of 44. And in the kids, it was not only lower than that, but it was different in the older kids. So the 8 to 12-year-olds had around half that much, and the younger kids had even lower still. So again, highlighting that if you're doing pediatric trials, you need to get the right age group. And what about the very young? So this is a study that was led by a number of researchers from UBC, showing that not only is the immune system very dynamic across the pediatric age range, but in the first week of life, it changes very quickly. And so these three graphs show changes in gene expression, changes in the proteins in the blood, in the proteome, and changes in metabolites in the blood happening just in the first week of life. In the red is the number of each of these things that goes up over this period of time, and in blue, the number of these different markers that goes down. So you can see, if you take the first one, over 1,000 genes where expression is significantly changing just in this 7-day period. So if you give an intervention to a 1-day-old baby, you may get a very different response to giving it to a 7-day-old baby. And if you're 25 years old, 7 days is neither here nor there. If you're a newborn baby, 7 days is huge. So I think it's really important that we think about, when we're doing trials in children, very, very closely think through the age groups and who the target population ultimately are going to be for any intervention. So I think we all know that in general, the future of our planet is with the children, but this has even been recognized within the ICH. And so this is a revision to the latest guidelines, so that they've recognized that targeted scientific and technical issues relevant to pediatric populations, regulatory requirements for pediatric study plans, and infrastructures for undertaking complex trials in pediatric populations has been considerably advanced in the last decade, without a parallel development of harmonized guidance. And so they've gone further to develop this new guideline and proposed it for harmonization of methodologies and strategies to incorporate pediatric extrapolation into overall drug development plans and speed access of new drugs for pediatric patients while limiting the number of children required for enrollment in clinical trials. So important recognition within the ICH of the relevance and importance of pediatrics, and the need for pediatric specific guidance. And you can't just apply the sort of kids are small adults philosophy. You need specific guidance to address the pediatric specific issues and people who have expertise in this area. And I just want to end with a warning, essentially. And so this came out from some work that was done around studies of vaccination and pregnancy, and lack of justified inclusion of pregnant women, but I think it applies equally to children and to other marginalized populations, and you end up in this vicious cycle. And what we really should be aiming for is that children are not unjustifiably excluded from participating in vaccine studies, which means that they then have the benefit from advances in vaccine technologies and are not left behind as new products are developed. And then they have access to safe and effective vaccines to prevent them and against emerging and re-emerging pathogenic threats. And so this is designed for pregnant women. This was focused on that, but I think it applies equally to children. And essentially the should be the basis should be of an assumption of inclusion rather than exclusion. That we should be thinking of reasons why children should be excluded rather than trying to think of reasons why they should be included, and inclusion should be the default. So I want to thank you for your time today. I'm happy to take any questions.
Good afternoon. My name is Gina Ogilvy, and I'm really pleased to be here at Clinical Trials BC Virtual Conference 2021 to speak about diversity in clinical trials and how we as scientists and clinicians and researchers can actually improve the diversity in participants in our critical clinical trials. So let's first start to talk about why diversity in clinical trials is important. We know that the foundation of evidence-based medicine and evidence-based practice is the clinical trial. A clinical trial obviously is where we randomly allocate an intervention to a group of people and randomly don't assign that intervention to another group of people. And this equal distribution of unknown confounders then allows clinicians to objectively confirm the impact of an intervention on an outcome. So it's been really critical as we move forward in evidence-based practice to have these clinical trials as the foundation of our care. But what we know is that increasingly there's concern about overall some of the limitations of clinical trials. One of the main concerns is that clinical trials are done in such a strict fashion that often those interventions aren't actually replicated in real life. And so that can limit the value and the validity of the findings to real practice in real life. And because clinical trials, particularly large clinical trials that are publicly funded, are the result of an investment by the entire public, it really is important for us to make sure that the findings have relevance broadly. There's also, though, emerging concern around the fact that clinical trials and the participants, particularly in clinical trials, do not mirror the populations where the results are intended to be implemented. And so then what happens is after all the effort to design and conduct and evaluate a clinical an intervention through a clinical trial, once all that hard work is done, it can then be actually difficult to extrapolate the findings from non-representative trial volunteers to a broader population. And so the risk there is that the findings from the trials do not apply to the entire populations, or trials may actually miss signals or findings that may benefit some different parts of the population. So this can then ultimately lead to a skewed or inaccurate body of knowledge upon which we base our interventions. This is becoming an increasingly important area of concern, but there was a very nice review by a Canadian investigator, Laurie et al., that was published recently that actually just looked at clinical trials, for instance, in oncology, and looked at 230 clinical trials that were done for the Food and Drug Administration. And what they found was that these trials, which actually influence policy and influence what medications were approved and weren't approved, only 63% of the trial included racial data. So even to understand whether there was diversity, we could just do it with just over half of the trials that were published. And in those trials where it was published, where racial background was published, Black and Hispanic patients were consistently underrepresented when compared with the expected proportion based on cancer incidence and mortality. In addition, they found disparities around things such as income, with higher income folks more represented in clinical trials than is in the general population. And I think this figure really shows it very well, where you saw that when you compare the incidence of the patients, of cancer in the patients who were in the trials versus actually who had the cancer, you can see that there was sort of equal representation for white participants. There was overrepresentation for Asian participants, but significant underrepresentation for both Black and Hispanic participants. So the question then for us as clinical researchers and trialists is, what are the challenges for diverse communities related to clinical trials? And certainly the base of it is lack of trust, which is due to previous mistreatment and previous experiences with medicine. There can be unintended language and cultural barriers in order to reach out to people and invite them to clinical trials, in order to make them aware of these opportunities and make them aware of how it can be beneficial for them and their community. There may be lack of awareness of the opportunity, both intentionally or unintentionally, where participants of different cultural communities just simply don't know the opportunity exists. And then certainly there's the consideration of costs. To participate in a clinical trial is inconvenient and costly for many folks, and that can include missing work, requiring flexibility with work, which if you have more shift work or more prescribed hours can be very difficult to do, costs of childcare and costs of transport. In addition, clinical trials often don't consider the different experiences of these communities, and so don't consider where and how the best places to recruit might be. It may be more appropriate to recruit in faith communities than it might in some cultural communities than it can for others. There can be inadequate attention to specific recruitment and specific retention efforts. So members of communities may join into a trial, but they're not able to stay due to other considerations that haven't been factored in when we're designing these trials. There's the accessibility of sites. Often our recruitment is on large university campuses or large hospital campuses, which can be actually quite remote for certain cultural communities. The frequency of visits can be very challenging, and also things such as consent forms and other trial expectations can actually be very intimidating for different cultural communities. The size, the length, the language within a consent form can be enough to actually dissuade folks from participating. So what can we do to improve participation of diverse communities in clinical trials? Well, I think the first thing we need to do is actually to make sure that we make diversity an expectation in trials, and that we look to our trials to represent our communities. And we also look to trials when they go to disseminate results to ask them about if they were representative, and if not, what efforts did they make to try to be representative, and how can they mitigate that going forward? And what might the implications of less diverse trial mean in terms of rolling out the findings in terms of policy? We need to provide clear expectations from funders regarding diversity to ensure that they are looking for their clinical trials to have diversity. And we need to implement monitoring on diversity in terms of clinical trial recruitment and enrollment. We also need to support researchers to authentically engage communities early, and to partner with community leadership to engage their communities in clinical trial participation. We need to ensure that trials align with the priorities for communities. And that's one very key way to get people to participate in a trial, is to make sure they understand how it's beneficial for them. But for us as research to also understand what are the key questions that community members are asking. Consider diverse sites for recruitment, including different community sites. So taking recruitment out of, say, large, academic health science settings and into the community. We need to look at decreasing some of the administrative barriers, things such as consent forms, locations that can be very intimidating for people. We need to look at budgets and ensure that we support researchers who ask for budgets for robust engagement. Robust and authentic engagement costs money, and it also costs time. And so we need to consider trial timelines and be reasonable, so that for folks who are trying to build those authentic engagements, that we give them time to do that, so that communities can feel safe and part of clinical trials. So I think this is a really important issue going forward. And as we go forward with designing and implementing clinical trials, we need to start to put diversity as one of the key elements in terms of trial design, both from the accountability end for the scientists and the researchers, but also for funders and communities. So thank you very much for this invitation to speak. And I look forward to discussing this topic on the panel. Thank you.
I'd like to thank the organizing committee for the opportunity to present this timely an important topic of ethnic factors in multi-regional clinical trials. More and more, we're moving towards a patient-centered care paradigm in which we're looking to apply the right medication to the right patient at the right time. At the basis of this is being able to get the data that helps the decision-making around what is that right medication and that right patient and the right time. Clinical trials are really the cornerstone of new drug approvals, as well as any applications for existing medications in a post-approval type of setting. We have to keep in mind whether we're planning or executing a clinical trial that without volunteers to participate in our clinical trials, none of this would be possible. Our current challenge is that clinical trials overwhelmingly fail to represent the demographic diversity of the populations that the drugs in development are aimed to serve. That's the topic we'll talk about today. Really, clinical trials currently are not enrolling representative populations. There was a Nature 2018 publication that reviewed clinical trials that were conducted over a 20-year period that were used for FDA approval. And what they found was that the median percentage of African American or African participants in a clinical trial ranged from 1.8 to 3.5%. Asian participants ranged from 0 to 7%. And any group that wasn't specified or described as white, black, or Asian was between 1.4 and 3.4%. We know just by looking at these numbers, these are not representative of the demographics in our communities. So the issue then is when trial participants are homogeneous, and this may be for a number of factors, whether it be gender, race, age, any of these factors, the findings may be skewed, and the result in that body of knowledge that's being generated is not generalizable to the general patient population. Now, having said that, there are some documents, some guidance documents that have been developed and can be applied to help us in our planning and execution of our clinical trials to overcome some of these lack of diversity issues. The first that I'd like to bring up is ICH E5, which is entitled Ethnic Factors in the Acceptability of Foreign and Clinical Trial Data. Now, this guidance document has been around for a number of years, but really at the core of it is really around how do we actually generate clinical trial data that can be used in more than one region or used for more than one registration of a medication in more than one region. It describes some of the regulatory strategies to minimize duplication of data collection and to facilitate the use of this data in multiple jurisdictions. It also describes the use of bridging studies in the event that we have discovered that our data that's been generated within a clinical trial is less diversified than required. It also describes some of the development strategies capable of characterizing ethnic factor influences that can affect our safety, efficacy, dosage and dosage regimen characterization. At the core of E5 is really summarized in this table. It talks about intrinsic and extrinsic factors. The intrinsic factors we often think about when designing our inclusion and exclusion criteria for our clinical trials. These may be genetic factors, age factors, gender, height and weight, maybe some predetermining conditions around liver, kidney, or cardiovascular function, some of the absorption, distribution, metabolism, or excretion factors that are often studied early in the drug development process. But often what's described here as intrinsic factors are not taken into consideration and need to start to be considered more and more in terms of how do we actually diversify our patient population. These include socioeconomic factors, educational status, language and language barriers, access to medical care or medical practitioners, how different communities look at disease and diagnoses and therapeutic approaches. All of these, what are considered here as extrinsic factors, become more and more important as we look at diversifying our patient populations. The second guidance document I'd like to bring your attention to is ICH E17, which is a more recent guidance document describing the principles for planning and design of multi-regional clinical trials. These are becoming more and more important as the cost of doing clinical trials increases and our ability to generalize the outcome from this data across multiple populations becomes more important. So some of the objectives of this guidance document is really around increased globalization of drug development. It It talks about the importance of gathering data from multiple regions and to be able to design our clinical trials to be able to be used in multiple regulatory jurisdictions. It describes some of the principles of planning and design of these multi-regional clinical trials, and it provides guidance on the use of this information in conjunction with other ICH guidelines. The scope of the document really is around clinical trials conducted in more region under a single protocol, keeping in mind the diversification that's required to be able to achieve that. It does describe a region as it could be a geographic region, it may be a country, or it may be a regulatory region. It talks about the relevancy of this document around early as well as later stage clinical development, although often it's thought about these larger multi-region clinical trials being done in later phases. There is a justification for use in earlier development. And it's intended for the use of the development and utilization of drugs and biological products. Some of the basic principles that are described here are really around how do we use these multi-regional clinical trials to increase the efficiency and decrease the cost of our drug development or post-marketing drug utilization clinical trials. The basic principle around a lot of the discussion within the document is that it must be planned early in the process. All of these topics that we'll discuss briefly here must be planned into a clinical trial and must be applied across the different regions and taken the multiple jurisdictions into consideration. It talks about the effect of applying across different target populations, and in particular taking into consideration the variability in the different regions in which you're looking to conduct the trial. It talks about a single primary analysis that can be acceptable to multiple regulatory authorities. And it talks about conducting this within other ICH guidance documents, in particular E6, which is the good clinical practice document, which helps to ensure and to achieve a high quality of data. There is extensive discussion around a lot of these implementation considerations that are listed here. Starting off with is study participant selection, and this is the basis of trying to get that diversification into the clinical trials. It talks about the selection of doses and how this may be different depending on the patient population you're targeting to include in the trial. It talks about the choice of endpoints and maybe the differential across the different regions that you're looking to incorporate into your study, and be able to take that into consideration early in the development of your program. It talks about sample sizing and the collection of the data to be able to use for both efficacy and safety information, and how differences across regions can lead to the different collection of that data, as well as utilization of some of that data has to be taken into consideration. The statistical analysis and selection of comparators may vary across the different regions in which you're looking to conduct a clinical trial, so it's important that you think about that and work that into the consideration of your clinical trial, as well as any concomitant medications that may be allowed or excluded, keeping in mind that they aren't always the same in all regions. There are many barriers to diversification that have been described in the literature or that we'll talk about here today. But there are many different reasons as to why minorities or different groups are underrepresented in clinical trials. One barrier is the obvious participation is the lack of trust because of past historical abuses. And I do think we have to keep that in mind as we're moving forward. Other barriers to participation can differ based on the patient population you're seeking to enroll, but I think there are some important ones for consideration. The first is language and cultural differences. Again, keeping in mind that not everybody perceives medical research or participation in a clinical trial the same. Health literacy and the ability to incorporate new information around healthcare and treatment is different in different patient populations. Religious or beliefs can have an influence over someone's willingness or want to participate in clinical trials. So we need to keep that in mind. And there's just a general lack of awareness and knowledge about what it does, what it means to participate in a clinical trial, and what's required to participate in a clinical trial. And I know Clinical Trials BC has done quite a bit of work to try and increase that awareness and increase the ability of patients to be able to feel comfortable participating in clinical trials. There are some extrinsic factors that need to be considered and planned into your clinical trial. Accessibility and clinical location is one. Often our clinical trials are being conducted at hospitals, which tend to be centrally located, but maybe the patient populations we're looking to target are not centrally located. So having access, easy access, is an important consideration. Frequency and time commitment of study visits is an important consideration, as well as transportation to and from. This isn't always easy, particularly if the patient populations are not immediately near where the clinic or site is located. Caregiver or family responsibilities have to be taken into consideration. Often people that are participating in these clinical trials have family responsibilities that have to be dealt with. There are economic factors, in particular when we're asking someone to participate in a clinical trial during the day, or maybe at a time in which they would normally be at work. There may be some economic factors that have to be taken into consideration. And then there's a general perception that minorities do not want to participate in clinical trials. Yet in reading some of the literature in preparation for today's discussion, often there's that perception that minorities don't want to participate. But there's also the other side of that is often minorities aren't asked to participate. So I think we need to be much more inclusive and be able to go out and do outreach to be able to ask for participation and work with the different groups to be able to include them as part of the planning and part of the process of the clinical trials. So in summary, we're moving more and more towards patient-centric care that requires more information, and really clinical trials is the ability to collect that information. That's the basis of our gathering and the basis of the interpretation of that data. Clinical trials continue to increase in complexity and cost, and I think that's where some of those multi-regional clinical trials or increasing the patient population to a more diverse and representative demographic of our communities can help drive down the cost of those clinical trials. And we have to think of complexity and working complexity out of our clinical trials to be able to make it easier for participants to want to participate in our trials. And then I've introduced you to a couple of the guidance documents that may help you in your planning and execution to allow for a greater diversity of patient populations, as well as looking across multiple regions and developing a multiple region clinical trial. And this is really going to set the direction for the next generation of clinical trials. I'd like to thank you for your attention today.
Good morning. It's a pleasure to be with you today for this important symposium on diversity in clinical trials. My focus will be on older adults in clinical trials. These are my disclosures. My plan is to address the following issues: characterization of research subjects by age, exclusions from study eligibility based solely on age, rationales for including older adults, evolution of the research funding environment, acceptable and unacceptable reasons for exclusion based on age, and finally, improving the clinical trial environment for older adults. When we turn to characterization of research subjects by age, we run the risk of ageism. So formerly benign terms such as elderly and old now take on a pejorative context, and the terms senior citizen and geriatric are suspect. Terms like elder and senior are still considered to be respectful, but the problem with all of these, of course, is that they are imprecise and subjective in nature. The simplest, most straightforward terminology is simple chronological age, age from the time of birth. And clinicaltrials.gov classifies clinical trials by those involving children, adults, and older adults, 65 plus being the standard criteria now employed internationally. Are older adults excluded from clinical trials? Yeah, they are. A systematic review of RCTs, which have been published between 1994 and 2006, showed that fully 39% of the trials excluded adults over the age of 65. When such a systematic review was repeated a few years later, the number of trials excluding adults over the age of 65 had dropped to 20%, but there were an additional 46% which, although they had no upper age limit, had eligibility criteria, which would be expected to disproportionately exclude older adults. Why would we want to include older adults in clinical trials? Well, the first reason is that chronological age is not equivalent to biological or functional age. Many adults in the age range 75 or 80 and above are fully functioning both mentally and physically. Their organ systems function normally. Other individuals in those age groups may be very different and experience a variety of dysfunctions, but simple categorization by chronological age is not an adequate criterion for exclusion from clinical trials. Next is the population prevalence of persons of older age. These are Canadian census data from 2016, focusing on the population aged 85 and older. At that time, there were 750,000 Canadians over the age of 85, fully 2.1% of the population. This is projected to grow sharply so that by 2051, there will be about 2.7 million Canadians over the age of 85, 5.7% of the projected population. There are many diseases and conditions that occur primarily in older adults. As well, there's the consideration of differential efficacy and safety in older age individuals. Atrial fibrillation is a good example of this phenomenon. In the general population, the prevalence of atrial fibrillation is about 1%. This rises to about 3 to 4% by over 60 years and fully 9 to 10% over the age of 80. Clearly, it would be nonsensical to conduct trials of atrial fibrillation without these older adults being included. Here's an example of an excellent trial. It's of a new anticoagulant, apixaban versus warfarin for the prevention of stroke and systemic embolism in patients with atrial fibrillation. The trial was sizable with 18,000 patients, and overall it showed a statistically significant 20% reduction in the risk of stroke or systemic embolism with the new anticoagulant. This trial had a very good representation of older adults. About a third of the subjects were under the age of 65, about a third 65 to 75, and fully a third over the age of 75. What we see is an interesting set of results. There's actually a trend against the new agent in the younger patients and a statistically significant benefit reduction of stroke or systemic embolus in the older age groups. Furthermore, because the control risk is higher in the older patients, the absolute risk reductions are even greater. Here we see in the 65 to 75-year-old group about 4 per thousand risk reduction, about 6 per thousand risk reduction in the older patients. The other side of the equation with oral anticoagulants, of course, is major bleeding. And here we see an important gradient. It's not quite statistically significant, but the older age group actually has more benefit in terms of reduction of major bleeding. And because their risk is higher in the control groups, we see a reduction of only 3 per thousand in those under 65, but fully 19 per thousand in the older age groups. So this dual benefit of reduced stroke and systemic embolism and reduced major bleeding is known to us only because the trials included older adults. And finally, there's the issue of equity. The Tri-Council policy statement of 2018 reminds us that equity requires distributing the benefits and the burdens of research participation in such a way that no segment of the population is denied the benefits of the knowledge generated, nor is unduly burdened by the harms of research. The NIH policy was adjusted in 2019 and framed in terms of inclusion across the lifespan. And they stated that older adults must be included in NIH conducted or supported human subjects research unless there is scientific or ethical reasons not to include them. The NIH policy document now addresses inclusion across the lifespan and specifically states what are acceptable reasons for excluding individuals based on age. And these include the disease or condition does not occur in the excluded age group, the knowledge being sought is already available in the excluded age group or will be obtained from an ongoing study, a separate age-specific study in the excluded age group may be warranted and preferable. The planned study will collect or analyze data on pre-enrolled study participants in another study from which older adults were excluded. There may be laws or regulations barring the inclusion of older adults, or it may be considered that the study poses an unacceptable risk to the excluded age group. The NIH policy document is explicit in regard to the fact that acceptable reasons for exclusion do not include cost, convenience, lack of explanation for age-based exclusion, concerns about noisy data. These are all problems for us as investigators. They make these studies more expensive, more complex, but they are not acceptable reasons for excluding older adults. NIH research applications must include plans for the inclusion of older adults or acceptable reasons to exclude them, and valid analysis of potential group differences. Reviewer worksheets require rating of the applications as unacceptable or acceptable before even being reviewed regarding plans for inclusion, accommodation, and valid analyses. The Tri-Council policy statement from 2018 on research involving elderly states elderly people shall not be inappropriately excluded from research solely on the basis of their age, and exclusion of the elderly shall not be based on easily remediable issues that are not germane to the research. So to the list of reasons to include older adults is funding agency requirements. Now we need some strategies for increasing recruitment, enrollment, retention of older adults in clinical trials. And the NIH workshop from 2020 is very helpful in this regard, suggesting robust and representative teams, intentionally inclusive plans with regular reviews, working with partners, including older adults themselves, the Canadian Association of Retired Persons, explicit budget items for dealing with the increased expected costs, minimizing staff turnover, accessing existing resources, taking lessons from prior successful trials, such as the SPRINT hypertension trial, explicit training of investigators and other personnel, and they've issued a 5T framework for recruiting older adults. So I hope in my comments I have been able to set out a clear picture of the issue of inclusion of older adults in clinical trials. The problems of excluding patients based on age alone, the rationales for including them, the evolution of the research funding environment to address these issues, acceptable and unacceptable reasons for exclusions based on age, and measures to improve the clinical trial environment for older adults.
Good afternoon. We are presenting today from the ancestral, traditional, and unceded territory of the Tsleil-Waututh. My name is Julia Bickford, and as Regional Director of Research, Evaluation, and Analytics at Northern Health, I have been tasked with building clinical research capacity and infrastructure in the North. I have the great privilege of working with Dr. Nadine Caron and Dr. Victor Daneko, my esteemed co-presenters today. None of us have any conflicts of interest to declare. Northern Health is geographically large, with about 600,000 square kilometers, or roughly the size of France. It serves over 300,000 residents. There are 54 First Nations in northern BC, and there are also at least 9 Métis chartered communities and many Indigenous people that live in urban areas. Clinical trials are very new territory for Northern Health and for the communities that we serve. We were asked to speak here today about diversity and inclusion in clinical trials. As a white settler having just arrived in northern British Columbia 10 months ago, I don't have the answers. I do have many questions. However, there is an important initiative in the North that has already paved the way for clinical research, and it's a model for how to move forward in a partnered way with communities. This initiative is the Northern Biobank, led by Dr. Nadine Caron. Dr. Caron and her research manager, Victor Daneko, kindly agreed to share their insights on diversity and inclusion. At this point, I'd like to pass it over to Dr. Daneko to provide more context of the diversity in Northern BC.
Unique geography and diverse natural environments of this vast landmass, which you were able to appreciate from the previous slide, are inseparably connected with its distinct demographics. As reported by Statistics Canada, roughly 13% of 5.1 million population of this province currently lives in rural and remote areas. Moreover, at present, Indigenous peoples constitute 5.4% of BC population and 4.9% of Canada's total population. These percentages might look like relatively insignificant numbers to you. However, when we consider the absolute numbers, it can be easily observed that we have over 660,000 of rural and remote dwellers in British Columbia alone. On top of that, we have close to 2 million Indigenous Canadians out of 38 million of total population. This is the real and appreciable size of population which is currently being underrepresented in modern cutting-edge biomedical research. And this poses an issue, an acute issue, I should say, that has to be addressed. It's worth noting here that this unique demographics is not distributed homogeneously across the province of British Columbia. For example, in Metro Vancouver, one can find only 2.3% of Indigenous citizens, whereas in place like Prince George, we have over 15% of Indigenous citizens and roughly 10% of minorities. Further north in city of Prince Rupert, these numbers are even higher. For example, the number of Indigenous dwellers in Prince Rupert reaches 40%. It also has to be emphasized that Indigenous people are very unique and highly diverse in their cultural practices, beliefs, and lives. Within geographical boundaries of Northern Health, one can find 54 different First Nations, each having its own unique way of life, its history, its worldviews. And it is anticipated and even expected that all future biomedical researchers are acutely aware of these cultural uniqueness and history of the population they are partnering with. The refusal to acknowledge the uniqueness of culture and traditions has led through the history to a great deal of problems and misdeeds, and I won't exaggerate when I say that such refusal has tarnished relationships between researchers and Indigenous peoples of Canada. And I hope that next presenter will elaborate in great depth on these problems.
Thank you, Victor. Thank you, Julia. So this is an exciting conference, and this is an exciting topic, and my thanks on behalf of our whole team for having us on this panel. There are many challenges with bringing research to the north, to rural and remote communities. And in particular, you'll notice that we have a particular focus on Indigenous populations and how we bring Indigenous voices to the table. As Victor said, there is a legacy of mistrust that has often resulted in hesitation of Indigenous peoples in Canada and really globally to participate in research. And this is because of some egregious research that's happened in the past, exploitive research that can be anything from culturally insensitive to downright disempowering, creating stereotypes, discrimination, often because the goal is to benefit mainstream science, commercialization, despite the cost that this might have on these communities and these populations. Sometimes we get tied up in the bars and the measurements that are used in Western medicine and in the world of academia, like number of grants, grant dollars, number of publications, number of podium presentations. And I think we need to reassess what we think is successful research. Part of this is also the disparities in education and the disparities in the roles of healthcare that we see when it comes to Indigenous voices. Indigenous people should be leading or partnering or consulting in all types of research, and that includes clinical trials. But there are challenges that are really across the board, not just with Indigenous populations, but across Northern BC, including particularly rural and remote communities across Northern Health. It's the geographic remoteness, low population densities, and we know this in clinical trials when you're trying to get the number of participants that you need in order to generate the power to come to the conclusions so that you can have the impact that you desire to help your community and the question at hand. Inclement weather conditions, perfect time of year for us to discuss that. Resource challenges, and I'm not just talking about human resources, which are definitely there, but also infrastructure resources that we have to deal with. It's hard to do a PET study. A study using PET scans. It's hard to do one that requires CT scans. In the north, in rural communities, remote communities, even lab values are sometimes hard to come by. And of course, there's the historical role of community hospitals and healthcare providers. For example, Julia brought up the Northern Biobank Initiative. When we started to bring this forward to primary care providers, to the lab in the hospital in Prince George, to the pathology department, a lot of the first responses were, that's why we left Vancouver or Toronto or Montreal. That's why we left our residencies and came up north. It's because we wanted to be community healthcare providers. We did not want to do research. We did not want to be part of academia. So part of the Northern Biobank Initiative was working with our partners and our colleagues to show that they can still be part of this research and critical to our team, yet not have to sit up at midnight and write the grants, that there's a role for everybody in this. And then there's the ripple effects of these challenges. If we don't address them, there's the inability to recruit a sufficient number of participants, which leads to frustration, which sometimes leads to the north being excluded. And then the other thing is the difficulty in managing trials and ensuring high quality work in distant locations. Sometimes just pure efficacy in terms of time it takes. There's high workforce turnover and staff shortages. And this isn't just about the healthcare providers that you hear that are often turning over, but also the ability to keep project managers or even students that then develop the capacity and subsequently get recruited down south or to metropolitan centers. There's a difficulty to maintain organizational commitment. And interestingly enough, this is something that is on the slide because we've heard about this, but I have to say the Northern Biobank is a project that has not suffered from that. We've had incredible support from Northern Health. And there's resistance to change, I think, because when it comes to research, we're all human and change can be challenging and change can be hard. There's also these challenges towards diversity and inclusion that I think we're suffering across the country, globally, across all areas of society, and research is no different. But research results without including these populations results in us relying on generalizability of the results. And then you get these results that are applicable to these metropolitan centers, and you hope that it works in your patient population or your communities, but those follow-up studies are rarely followed through. So it may say in a paper or a manuscript that that's the area of future research, and that future research is never completed. So we need inclusive recruitment of Indigenous, rural, remote, and northern communities. to be part of the research process, and to be participants in the research that's being done.
So Dr. Caron, from your experiences, what does appropriate community engagement look like as we try to build capacity and infrastructure for clinical trials in Northern British Columbia?
It's a great question, Julia, and certainly one I know that you and I have talked about with our NBI team and my colleagues here. I want to recognize also a colleague, Dr. Mek Chongo, who is a big part of the Northern Biobank Initiative and other projects that we're doing, and has helped us with our presentation here today. I think you have to sort of take that question, and I'll put it right back in terms of, here's a bunch of questions that you need to have the answer to in order for your community engagement to be appropriate. I think first of all is, what are you asking? When you go to engage with these community members or community leaders, what are you asking? For example, if you have a research question, it's going to be really important to recognize, is that a priority for the community? Is it something that they're interested in? Is it something that, while it may be really paramount in your world, your area of study, your area of expertise, if it's not a priority in the community you're reaching out for, brush your ego off. Realize they're not turning down you, they're not turning down a partnership with you, and they're not even turning down the research project. It just might not be the right time or the right place for that. And in a time of COVID, more than anything, be patient and know, what are you asking for? And sometimes it's just, what do you think? And then another question, when do you ask it? We've heard the term community-based participatory research, but really, when do you ask it? Is as soon as you start to have ideas. It's good to reach out to these people that you want to work with, that you are aiming to partner with, that you want to build these academic relationships with. So for example, with an NBI or the Northern Biobank Initiative, we had community engagement for about 2 years before we even thought of applying for a grant. Because we needed to know, like, what does a biobank in the North really mean? What's a biobank for First Nations communities really mean? And if they said, we don't care, we don't want a biobank, well, there'd be no reason to even apply for the grant, let alone get our all our efforts in place and have every academic supporting us down in Vancouver when the communities up in the North didn't need it. And luckily, we got the absolute opposite response. People were really thrilled, and they were part of it right from the beginning. Who do you approach is an important one. And this is recognizing that when you're going to have community engagement, it depends on the research question. It depends on the community. And it's important to recognize that it depends it's really timely, because who you approach for one project might be someone completely different for another project. And part of that is recognizing that when you're building the process, putting out the research question and asking people what they think, you will hear from the community who's important to have at the table. And if you involve and you're inclusive, that's going to lead you to having voices and perspectives to help you really build the project from the ground up with a methodology that's going to fit that community. Where does this engagement occur? A little bit hard during COVID, but I think it's really important to recognize that it's hard to say that you're working on a rural project when you're in a city. It's hard to say that you're really interested in Northern health and you're going to do this Northern project when you're in the South. It's really hard to say that you're going to do this project on Indigenous perspectives in cancer care or in an Indigenous biobank if you're not Indigenous. It doesn't mean you can't be part of that. It doesn't mean you can't be part of the solution, but it does mean that you need to round out your team in order to have that perspective at the table. And that means going into those communities so you can learn from them on the ground about what they're working with. How long does this take? This is a whole talk unto itself. The main thing is, how long does community engagement take? Well, from the NBI perspective, it started before the project even began, and it will continue well after the project is over. A colleague of mine, Dr. Laura Arbour, calls this the conversation that never ends, particularly when it comes to Indigenous populations. Expect your Gantt chart to look weird. Expect that the consultation starts before time zero and it goes off the end of the chart, and expect to have to explain that to funding agencies where it doesn't really fit their reporting model. Why does it matter to you as a researcher? I think this may be the most important question. If you want a community engagement and you want it to go well and you want it to matter, then essentially the main thing here is people see what you're there for. And if you're there for the right reasons, that you want this research to have an impact, that you want to improve the health and wellness of whatever population you're working with, then if that's what matters to you the most, then you're going to have a successful partnership and you're going to move forward in a good way. I think overall community engagement, to be appropriate, needs to be responsive, meaningful, respectful, incredibly flexible. Learning should be reciprocal. So for the most part, you're not just there as a researcher to build capacity. Your capacity will be built by reaching out and going to places that you haven't been and working with people that you haven't had the opportunity to engage with. And it's not enough to be cognizant of diversity anymore. It's not just cultural safety and cultural humility anymore. It's really the cultural security. It's something bigger than just being aware of diversity. And that's a whole other topic that maybe we'll touch on in the Q&A. The process design for these clinical trials should allow for a broadening of the eligibility criteria, which I think is why we're here, which is why I'm so happy that Dr. Stewart asked this perspective of rural, remote, and Northern Indigenous populations to be here. Make it easier to participate in these trials. In some of the trials that I've had tried to get patients of mine involved in the area of cancer, for example, it's been incredibly challenging because of the amount of travel that they need to do in order to be involved, as opposed to when I was a resident in Vancouver, where my patients just had to walk down the hallway, or the actual phlebotomist would come to them while they were sitting in the clinic. Remember that if you're going to involve Indigenous communities or northern, rural, remote communities, you need to budget accordingly, because that will take time, it will take travel, it will take accommodation, and it will involve also making it so that you can facilitate that for the participants and recognize that it costs them to travel. Someone in Vancouver, it may take them an hour to do that part of the research project. For someone traveling, it might take them 3 hours to get to Terrace or to Fort St. John or to Prince George. Are you going to compensate them for the 3 hours in, the 3 hours out? Because that's a lot of time. And these are people with that their time is worth something, and budget for it. And involve communities in various areas, recognizing that diversity includes language, land, culture, and make sure that you have those processes in place.
Thank you so much, Dr. Nadine Caron. Those are really great insights on community engagement. This is a real culture change for us at Northern Health, and I think there's also a need, in addition to engaging communities, a need to engage with healthcare providers who, as you talked about previously, may not see themselves as academics or research oriented. So from your perspective, how can we move forward with clinical research in a way that's inclusive of clinicians in rural and remote environments?
You know, another great question. I know we're getting short on time, and I think we're going to have a really robust Q&A. So for the most part, recognizing that healthcare providers have to start somewhere, start it in their training. So recognizing as they're kind of moving through the process of undergrad into medical school, residency, and the comparable areas of training for other healthcare providers, make sure you give them opportunities in research and show them the impact that it can have. Teach the importance of evidence-based medicine. The decisions we're making today are based on research. The only thing is that the decisions I'm making for my Indigenous communities and populations and patients is not based on data that's come from them necessarily. So recognizing that. Recruit accordingly and collaborate with community so that you can recruit academic physicians or people who want to be part of this process more actively into places like the Northern Medical Program. So on behalf, I guess, of our whole team, not just Julia, Victor, Mek, and I, but from the whole team that's growing within Northern Health,
So thank you very much. Can you hear me now? Yes, thank you. So thank you to all of the speakers, and I think you've done a splendid job. The issue of diversity in clinical trials is almost like a diamond with multiple facets, and everybody's looked at this through a different lens or a different facet. And I think it's provided an extremely rich articulation of the issues that we're trying to deal with. So I would ask people to please use the live Q&A session if you have any questions. And I'm going to start by looking at what's been upticked here. And this is a question for Gina Ogilvy. And what would ideal monitoring of diversity in recruitment look like in terms of transparency and reporting? Gina?
Yeah, thanks for the question and the opportunity. I think what we have to do, often this type of sort of setting expectations starts at the level of the granting agency. So what we don't want to do is have challenges where all of a sudden research grinds to a halt because people aren't used to doing this. So ideally, of course, we're going to start with a process of consultation and get input from researchers and scientists and clinicians about how this could be done. I think the first thing we should do is just make an expectation that you need to gather cultural, race-based data, ethnic minority, and indigenous status. And CIHI has actually come out with some preliminary guidance on this, and they're actually looking for feedback. So I think that's the first thing, is we just say, you know what, as part of recruiting, you need to get this data. Because it was a bit surprising to me that in that review that was conducted, that almost 40% didn't even report that data. And so to me, that would be the first part. Then what we can start to do is say, and I think the speaker who talked about looking at elder citizens, that's the kind of thing we need to say is, how are you working to make sure that everyone has an opportunity? And that's the type of monitoring I think that we want to embark on. And that's the kind of standards we need to just start to articulate. And then we can start to hold or make expectations and start to structure accountabilities as we go forward.
So thank you very much. And maybe just another question to Dr. Cairns. John, you were talking about elderly and clinical trials and inclusion and so on. Could you offer any comment on sort of the phases of clinical trials, whether phase 1, 2, and with potentially toxic interventions, how one accommodates elderly in those studies?
Yeah, that's an interesting question, Gavin. I haven't thought a lot about that. We're usually looking at the issue of trying to have population representativeness. I think phase 1, we're more concerned with can we safely give this agent? And as we go into phase 2, what sort of doses might be appropriate? What kind of effects might we have, both not so much in efficacy, but in terms of toxicities. Older adults traditionally haven't been part of that. The representation has tended to be younger. It's a very difficult question. I think probably the place you want to start with is people where you have the expectation that their systems are working normally. Those are usually in younger people, and that any toxicity is going to have the least likelihood to be dangerous. I'm not sure how much we're likely to move towards trying to have representativeness across at least age groups in terms of phase 1. We probably want to make sure we've got representation according to various levels of organ function rather than age as a criterion. But it's an interesting question. I'm not sure what the right answer is in regard to phase 1. It's a very good question.
Okay, thank you. And maybe to direct this next one to Jim Pankovich. And Jim, you were talking about the ICH E5 and E17, but somebody has asked here if you could comment on, there's kind of two issues. One is fairness and who gets to participate in multi-regional trials. And secondly is, does the lack of diversity risk affecting the validity of the trial outcome? Can you comment on that balance?
No, it's a good question. So maybe to answer the second one first around the validity of the data. I don't think it affects the validity of the data per se. What I think it does is it affects the generalizability of the data. And we've often in the past have said, okay, it worked in this clinical trial, so it should work in the entire community. And I'm not sure, particularly with a lot of the new, therapies that are being developed, that's really true. So I think we need to increase that diversity to be able to generalize the data. So I think the validity of the study itself is always based on the participants who are actually enrolled and partake in the treatment. I think that's where we really can rely on doing these clinical trials. It is a challenge to recruit a diverse population, and I think a lot of the speakers talked about that today. It does take extra planning. It will take extra resources, but it also, one of the things that was talked about was engagement. And I think often we kind of just come in and we try and engage on a study-by-study basis. I think we need to take a more systematic approach to that and have continuous engagement, not on a study-by-study basis, but on research in general.
Okay, thank you. And here's an interesting question, and maybe I'll just leave it to whoever wants to step up first. That the question is, how big a change is in current practice is required to realize these diversity goals? That is, if we did the clinical trials research right, and if it truly was equitable, how much would our current practices have to change? Any takers?
Older adult. Is my speaker on? My mic?
Yes. Yes.
It actually has changed quite dramatically in the last 20 years. And I pointed out in randomized trial overviews looking at, I remember when I was first doing large clinical trials, we frequently had age criteria for exclusion. We almost never have that anymore. We exclude people for organ dysfunction for good reason, but we've come to recognize that age alone is not an appropriate criterion for exclusion. It's not operative, and it also has problems of non-generalizability. So this is not new. It's evolved quite dramatically in the last 20 years to a more appropriate approach.
Okay.
Yep.
And go ahead, Nadine, and then I have a question for you.
Sure. I think it's important to see, I can see the age part, and I think some populations are lagging behind that. And I think I like the idea of not doing it project, but doing it by an entire process. And I think the process that academics, which are researchers, pharmaceutical companies and industries which do research, I think where they respond is where basically the process is. I think REB should be responsible for assessing this so that you have to explain why your study isn't doing that. Granting agencies should be looking at that, just like the NIH and the CIHR and the Tri-Councils are doing. I think that publishers should be looking at that. They should not publish something if they don't have a rationale for why there has been an exclusion of a certain population. And I think the last one is the most important one because it's the one that's been left untouched. I think publishers are somehow get away scot-free. And in the indigenous area, particularly in genomics, what we have been working on are with publishers and trying to hold them accountable and to see if we can get publishers like Nature to come on board and actually be a leader to sort of say, if you submit something and you show that you haven't done proper engagement, that you haven't done things in a safe, culturally appropriate way, that things won't be published. And I think then the rubber will hit the road and researchers will realize that they're in a space that's no longer negotiable.
Okay. Thank you, Nadine. And while you have the microphone there, a question that is asked of yourself that recognize that clinical trials are sponsored both by academic sponsors and biopharma sponsors who are developing new drugs and biologics. Can you comment on engaging industry sponsors in these important discussions, particularly through the lens of Northern Health Research or through your view?
Right. Northern Health is a little bit different because I think it's a population that's geographically isolated and has challenges that way. To answer that more through an Indigenous lens, I think it's actually a very easy question. It's actually not an answer that I can give a potential funder from industry or the pharmaceutical companies. It's actually a question that should be brought to the communities, to the leaders, to the Indigenous organizations, whoever is at the table and part of the research, so you can ask them what they want. And that's why if you do the engagement early and you set up a process and you have inclusion, not only in the participants, but in the actual team who are doing the research, you can have a very comfortable, open conversation about what is reasonable, what is expected, what is needed in order to enter a partnership in that way. And it's not the researcher. It's actually a very to be in, but very responsible space to make sure that you are humble and you realize that you don't have the right to give that answer.
Okay, and Manisha, I'll maybe start with yourself, because there's an interesting question here about gender diversity, and then it extends to ask about pediatric trials and gender diversity. How is that accommodated, or how much is it accommodated?
Yeah, so I mean, I have to say for pediatric trials, unless we're specifically targeting a single sex, for example, if, studies we've done in the past with Gina Ogilvy on human papillomavirus vaccine, when it wasn't given to boys, clearly that was just a single sex trial. But in the most part, I think that a lot of the recruitment strategies do accommodate both male and female. And I think if anything, certainly when we get into the older adolescent populations and even the adult populations, we find it's often the other way around, and we have overrepresentation of anything of females in some of the research studies that we do rather than males specifically. And it's something we keep an eye on, and up to now has not been a major issue.
Gina, any comments there?
That, I think the question is, we now routinely look at both sex and gender, and unless there's a good reason, so for instance, we would be interested, if say in some of the work that I collaborate with Manish on, we are interested in doing cervical cancer screening. So the individual has to have a cervix. So we would be interested in that that sort of biological issue, whether you have a cervix or not, because we can't screen without it. But other than that, I would agree with Manish that in most cases, we have our studies are open to all genders.
Okay. And maybe, Jim, this is a question for yourself. I mean, could you see, what role do you think Health Canada plays in terms of putting more parameters in place to see a shift in trial recruitment towards more inclusive studies?
Well, I think more and more the emphasis could be on some of the guidances and the regulations to, in the approval process, to better understand more of the populations in which the drugs or the therapies may be utilized. And most of the regulators have taken a more traditional approach. You do the research, you apply to the regulators for approval to use the drug, and they'll evaluate the data that's in there. I think more and more they should be looking at earlier in the development process. We often talk about, okay, hepatically impaired or renally impaired or other differentiation factors. Maybe we need to actually start talking about more of what some of the regulations call those extrinsic factors, and say, okay, if we want generalizability of the data, we want to be more inclusive of those types of factors as well.
Okay. And maybe, this is, I'm glad the audience are asking the right questions. And maybe, Julia, I'll ask you to start, but somebody here is asking, what are the best resources available and the best practices to ask questions and collect information about diversity in research study, asking about ethnicity, gender identity, Indigenous status? Do you have a comment on that, about what are the resources that you'd recommend to colleagues?
I think first and foremost, can you hear me okay?
Yes.
First and foremost, I think is to make sure that researchers are partnering with people in the communities that they want to do research with. So that's, I would recommend that as the #1 resource, is getting those communities on board in your study and learning about the resources there. I also know that health authorities, we all have resources. We have, for example, with Indigenous research, we have Indigenous health teams within the health authorities that can also help direct you, and then our academic partners, in the university at UNBC, for example, there's Indigenous centers and academics who can help connect you as well. But Nadine, you may also have other ideas.
I think you covered that really, really well, Julia. I think the most important thing is the dialogue here, and just looking at the number of participants and that have come to listen to this forum, I think is great. Knowing that this is a question to ask. Researchers, academics, leaders, people with passion to have a question. Generally, I think you're all pretty good at finding the answers. It's knowing that you have to ask the question now, I think, is the success of the panel.
And maybe as we're getting close to time, I'll just sort of a more general question for the panel. Language and cultural differences have been challenges in many trials with complex behavior change interventions. And there's usually a very limited budget for translation of material or to hire multiple interventionists with different language skills. Any comments on supports that researchers and clinicians, clinical investigators should turn to as being helpful?
Yeah, Manish, I can start, and then, sorry, Jim, I can start, and then we can jump to you. I think it's such a good question. I remember when we started recruiting for one of our big trials, we were not able to actually retain, according to the institutional hiring practices, we weren't able to select a candidate who could speak Mandarin. So that was a challenge for us. So we had lots of interested participants who spoke Mandarin, but we didn't have someone who could speak to them. So we need change at institutional levels to understand this is a priority. We need to think about things like even consent forms. When you hand someone these 8, 10, 12-page consents, that's enough to turn them off. They don't know what they're signing. So we need to view trimmed down, streamlined consent forms as actually being more participant-centered. We need to start to really make the expectations at the level of the institution, and that the institution takes on some risk and also some priorities. So for instance, even budgeting and timelines for a trial. As Nadine said, you shouldn't be, getting your funding and then starting the engagement. So hopefully you've started well before. But even with that, given the time it takes to get funding, to maintain that relationship and have an authentic relationship, that just takes time. So maybe our 3-5 year grant cycles are not appropriately community and participant centered. So I think there's a lot of work that we need to do to actually just change the standards and expectations and start to, and that's why a session like this is so important, so that when you are putting a budget.
I may just cut you off there, Gina, because we're going to run out of time here. Obviously a number of questions, and I just love the richness of the conversation that we're having. I am going to say thank you to all of the speakers, as well as thank you to the audience for participating. Please, after the session, feel free to visit the exhibitors hall. The team has put this together, and I think this was a great conversation about diversity in clinical trials. Thank you to all of you.
Pleasure. Thank you.
Thank you.